Aseptic technique is a group of behaviors and techniques that prevent microorganisms from getting into spaces. In the microbiology laboratory, we will be using aseptic technique to prevent microorganisms from getting into our cultures. In healthcare, you use aseptic technique to prevent microorganisms from getting into your patient. In the microbiology laboratory, there are typically two types of cultures you work with. The first type is a pure culture. Pure cultures are one kind of organism, easy to identify. Mixed cultures will have two or more organisms in them, but the organisms will be unique and easy to separate using standard laboratory procedures. Contaminants are the unwanted organisms. These organisms can be difficult to identify, and you want to prevent contaminants from getting into your cultures by using aseptic technique. Microorganisms are absolutely everywhere. That is what the word ubiquitous means. Aseptic technique helps prevent contamination, and in the microbiology laboratory is a method of handling your cultures to prevent the introduction of environmental organisms. Some of the sources of contamination include air currents, the use of non-sterile equipment, and contact with the environment. Microorganisms are in the air, so we keep doors and windows closed to minimize those air currents. Occasionally, methods we use in the laboratory will create aerosols. These are fine sprays of microorganisms into the air. These organisms may stay in the air momentarily, but they tend to settle onto surfaces and to the dust on surfaces. Air currents and rapid movements, which can stir up dust and air currents, are ways to reintroduce those organisms back into the air and have them resettle someplace else. So we want to make sure that we keep any kind of air current to a minimum. Also, you constantly shed skin cells and microorganisms. For that reason, you want to never reach over a sterile area or any of your cultures. To make sure that your equipment is sterile, you're going to sterilize your loops and your needles in the Bacticinerator. The core of the Bacticinerator gets to 1500 degrees Fahrenheit, so this will quickly sterilize your wire. You should watch it glow red for 3 seconds. It is very poor technique for you to prop the loop or the needle in the opening of the Bacticinerator. For one thing, it's just poor technique. The aluminum handle will transfer heat, and when you go to pull it out, you may burn your fingers, causing you to drop the loop or the needle, which will contaminate it. The other thing is that 1500 degrees Fahrenheit will melt aluminum, and one of the things that can happen if you prop your loop or your needle in the incinerator is the handle may melt, leaving the wire part in the incinerator and the handle laying on your table. We use Petri dishes to work with our organisms, and it appears that we always work with them upside down, and that's true. The lid of the Petri dish is a little larger in diameter than the base, and the base is where we pour the agar. We want the agar plates to always be upside down. There are two reasons for this. One reason is that when you take the plates from the warm incubator environment and bring them into the cooler air environment of the laboratory, condensation may form on the lid. If you had the plate sitting with the lid up, that condensation, that water could develop or pool into one area and drip onto the surface of your plate. If microorganisms get into a pool of water, they can all swim together, so you would lose any isolated colonies you might have had. The other reason is it's just easier to handle the plates this way. When you work with your microorganisms on the plate, you want to minimize any kind of handling of the plate. If you have the agar side up, it's very easy to simply lift the bottom out of the lid, leave the lid sitting on the table, it was sitting on the table anyway, and then you can work with the organisms on the plate. If you were to do it the other way, have the lid up, you would have to take the lid off and set it on the table, and then you would have to figure out how to pick up that agar piece without sticking your fingers in it. So this is just an easier way to handle the plates. Also, when you lift your plate out, you want to make sure that your agar surface stays sort of parallel to your work surface. This means that there won't be as great a likelihood for the organisms to drop onto the surface of the plate. When you are holding your loop or your needle, you hold it somewhat like a pencil. You want to hold it in your dominant hand, prop it between your first two fingers, and hold it in place with your thumb. Sometimes you will have to remove lids from tubes. You can't take the lids off and lay them down on the countertop. You have to learn how to use that hand that's holding the loop, wrap your little pinky finger around the lid, and either pull the tube off and leave the lid in your hand or unscrew the tube if it's a screw cap. But the lids from the tubes cannot be set on the table. They must stay in your hand. You want to handle cultures in order to prevent contamination. You never talk to your cultures. Now, you'll be very chatty in lab and you'll be talking with your lab partner, but when your plates are up or when you're working with your microorganisms, you should be quiet. You should not be talking to your partner. If you're ever not sure if something is sterile, then you should assume that it is not sterile and treat it accordingly. That means you should discard it or start over.